In this lab topic, we are going to learn how to generate and interpret an ANOVA in Jamovi, also create a graph illustrating the results of an ANOVA. In this lab topic, we are going to learn about connecting an ANOVA in Jamovi. So first we'll learn how to generate and interpret an ANOVA. Then we will learn how to create a graph illustrating the results. And finally, learn how to write up the results of an ANOVA in APA style. Hopefully this format is starting to feel pretty familiar to you. We've been doing kind of the same sequence of events for all of the different hypothesis tests we have been learning how to do in Jamovi in these lab videos. Just as a quick review, recall from lecture that we use an ANOVA when we are comparing the means of more than two populations. Right, so we have more than two samples, each from different populations, we're comparing their means, that's when we use an ANOVA. And here I just want to note that I have put kind of the word one way before ANOVA because I just want to let you know that what we're doing is actually a specific kind of ANOVA called a one-way ANOVA. There's actually many different types of ANOVAs, and you will encounter these other kinds of ANOVAs in more advanced stats classes. But what we cover in intro stats is typically just the one-way ANOVA. And I'm just going to call it ANOVA for the sake of brevity for this class. But I just did want to mention that there's many different types of ANOVAs. So we are going to work in Jamovi to see how we can answer the question of whether the difference between sample means is significant. For the example in this video, we are going to do a little hypothetical experiment. So there's a hypothesis in cognitive psychology that memory may be enhanced by color. And also the more highly saturated the color, the more pronounced the effect. So say I want to test this hypothesis. Say I gathered 150 participants and they were randomly assigned to view one of three versions of the same picture. Either the picture in black and white, the picture in low saturation color, or the picture in high saturation color. Then after viewing the picture, they were asked to write down all the items they could remember from the picture. And then it was recorded how many items they remembered correctly from this picture. So I just want to make this a little bit more concrete about what this could have actually looked like. So I have kind of three versions. Essentially, the participants would be doing one of three possible versions of this experiment. All right, so this would be the first screen they would see in the experiment, and they would be told to study this image for for a certain amount of time, say a couple of minutes. And so this would be what participants in the black and white group would see. This would be what participants who were assigned to the low saturation color group would see. And this would be what participants assigned to the high saturation color group would see. Okay, so they would be told to study the image, and then the next thing they would do would be to list all of the items that they could remember in the picture. Right, so they'd be given time to list everything that they could remember, and then the experimenter would write down the number of items they correctly remembered. So they would kind of mark this person's response, and here would be where they indicated how many items they remembered correctly from the picture. Right, so that is what this little experiment would look like. Okay. And the data from this hypothetical experiment is posted in the data set for this lab. Okay, so here is the data set, and you can see that there's two variables in this data set. The first is the number of items correctly remembered. Okay, so for each person, this is the number of items that they correctly remembered from the picture. And the next variable here is the picture viewed. So this is just the variable that tells you for each person whether they were in the high sat, low sat, or black and white group. All right. So how do we generate and interpret an ANOVA in Jamovi? And I'm going to walk you through the steps here. So we go to Analyses, ANOVA, and ANOVA. Just another note here. So notice that it says like it's got like one-way ANOVA and ANOVA. The one-way ANOVA will give you the same result as what we're doing here. It's just that if you click ANOVA in Jamovi instead of one-way ANOVA, it just gives you a little bit more options for information. And so we could go here, but I'm going to have you go to ANOVA rather than just one-way ANOVA, like I said, because we just get a few more statistics in this window here. Okay, so don't be confused. This also gives you the results of a one-way ANOVA. And this is just what I'm going to pick for you to do here. All right, so you're going to move the dependent variable, which is the number of items remembered, into the dependent variable window. And fixed factor is the grouping variable, and you're going to move picture viewed into the fixed factors window. Under effect size, you are going to click off eta squared. So in lecture, I said that the effect size for Jamovi is sometimes referred to as r squared, and sometimes it's referred to as eta squared. So in the textbook, they refer to this as r squared. In Jamovi, they call it eta squared, but it is the same, and you would interpret it the same. Just they call it different things, which is so fun. So, okay, effect size, under assumption checks, you are going to check off homogeneity and normality tests. Under post hoc tests, you are going to put picture viewed into this section. So there's two windows here. You're going to put picture viewed into the second window. And then right under here, you can see that there's a list of correction. The correction, I think the default is that Tukey is clicked off. And if this is clicked off, then it gives you, it's going to give you this table in your output that tells you which groups differed significantly from which of the other groups, right? It'll compare the groups individually. And if you click Tukey, it will have the p-value adjusted for number of comparisons and maintain an overall 0.05 significance level. So you want to make sure Tukey is checked off. And we can click off Cohen's d for effect size. Okay, so that's going to give us an ANOVA. One thing that this window does not give us, which I wish it did, is it doesn't give us the means for the groups separated. So it doesn't give us, for example, the mean number of items remembered for the black and white group, the high sat and the low sat group separated. So, and we want that information. We need that information to interpret the results. So we're going to go and get that, and we are going to do that by going to Exploration, Descriptive Statistics, and just ask for the number of items correctly remembered split by picture viewed. And you just need the mean and standard deviation and n as well. I guess you want to have that. So just to condense your chart, we can just have this information, and that is all we need. So here we have our output, and what I have just generated in Jamovi here will look exactly the same as what is on your lab handout. So I'm just going to shut down Jamovi and exclusively refer to the handout here. And so here is the ANOVA output. A lot of information. As always, it's annotated, so you know exactly what every important number means. And I encourage you to kind of to go through this so that you understand. But what we're going to focus on in the video here is interpreting the results. Okay. And so the first thing that we are going to do is just look at the means for each sample. Okay. We're going to look at the means for each sample and just note the differences. All right. So this is an annotated output number 9. And we're just looking at the means for the high sat, low sat, and the black and white groups. this tells us that in the high SAT group, on average, participants remembered 9.08 items correctly, whereas in the low SAT group, they remembered 5.52 items, and in the black and white group, they remembered 5.30 items. Okay, so we know that there appears to be some differences between group means, particularly in the high SAT group. So we're just going to kind of make note of that. And then the next thing that we're going to do is we're going to look at the p-value, and this is the p-value for the f-ratio. So kind of behind the scenes, Jamovi is calculating that f-ratio based on the sample information. The p-value is specifically for the f-ratio, and just like we did in lecture. And this is going to allow us to determine if the null hypothesis should be rejected. As always, if the p-value is less than the significance level, that tells us that we should reject the null and results are significant. Okay, so let's take a peek at the p-value for the f-ratio. And you can see that it is significant, right? We've encountered this before. If the p-value is really, really low, Jamovi won't give you a number. It will just tell you that it is less than 0.001. So we know that our results are significant, that there is a significant difference between at least two of the groups. As we saw in lecture, though, just seeing the p-value and noting that it is significant doesn't tell us which groups' means are significantly different. So for this, we need to look at the post hoc tests. We saw an example of post hoc tests in the lecture example that I did. So this table we have encountered before in the lecture example, and this is what we always look at when we get a significant f-ratio to see which groups differ. All right, so as I did in lecture, I'll kind of go through this with you again. But I just also want to note here that I have written out the kind of steps for examining the post hoc tests. If you would rather look at this, then go back to the video when you are working on your assignment. Okay, so but essentially it's pretty simple. Each row presents the comparison for two of the groups. So the first row here, this Jamovi is doing a test to compare the high SAT group to the low SAT group. Essentially, you just want to look at the mean difference column and then the p-Tukey column. Right, so mean difference tells us because it's high SAT minus low SAT, this means that the high SAT group remembered on average 3.56 items more than the low SAT group. And then we jump over to the p-Tukey column, which is this adjusted p-value. So it's adjusting for multiple comparisons so that the overall comparison is kept at, the overall significance level, is kept at 0.05. And we can see that this is a significant comparison. So the high SAT group remembered significantly more items than the low SAT group. And then we have the second comparison here, which anytime you see kind of a blank, you can assume it is the same as the cell above. So this row here is comparing the high SAT group with the black and white group. The mean difference is 3.78. So that means the high SAT group remembered 3.78 items more on average than the black and white group. And then we skip to the p-Tukey column and see that again, this mean difference is significant. So the high SAT group also remembered significantly more items than the black and white group. This next row compares the low SAT group with the black and white group. And the mean difference is 0.220. So the low SAT group remembered 0.220 more items on average than the black and white group. If we skip to the p-Tukey column, we can see that this p-value is much greater than 0.05. And so we would say that this mean difference between groups, the low SAT and the black and white group, is not significant. All right, so that's the next step in interpreting an ANOVA. So we know now, we can kind of conclude that yes, there was a significant difference, and the group that was remembering significantly more was the high SAT group. So the high SAT group remembered significantly more than the black and white or the low SAT group, but the black and white and low SAT groups did not differ significantly from each other. All right, so the next thing that we do is look at the effect size, which is eta squared, can be found on annotated output number 5. So we scroll up there. Here's the effect size. So this tells us that Our effect size was 0.412, so we can interpret that as 41.2% of the variance in the number of items correctly remembered, right, because that's the dependent variable, can be explained by the variable that divides up the groups. So that is the picture that they viewed, right, whether it was the black and white, high sat, or the low sat group. So we want to always just note the effect size. And then the final step is checking to make sure that the assumptions are met, which we can find in on the output number 6 and 7. Scroll up, 6 and 7. Again, this is hopefully feeling fairly familiar. We've done this multiple times before. We have two tables, one for each of the assumptions, and we want to look at the p-value for each of these assumptions. So for the homogeneity of variances, we can look at the first row. We can see that Levine's p-value is 0.004. So remember that in these situations, we want the p-value to be greater than 0.05. If we have a low p-value, less than 0.05, that means that that assumption was violated, not met. So we know then from looking at this p-value that the assumption of homogeneity variances was violated. We're not going to do too much more with violated assumptions at this point. In general, it's thought that the tests are robust to these violations, particularly when you have a higher n. So, in this case, we're just going to kind of make note of that, but assume that the test is fairly robust to this violation. And so we're going to just proceed with the analysis. The next assumption is the normality assumption. And again, you can just look at the Shapiro-Wilk row, so the first row, take a look at its p-value. In this case, the p-value is greater than 0.05, so we have like a non-significant p-value. That's what we want. That tells us that the assumption of normality has been met. Okay, so that is how you interpret an ANOVA. Right, so you're looking at the means, looking at the p-value. If you have a significant p-value, you want to interpret the post hoc test table, look at the effect size to get a sense of the magnitude of the difference, and then also check to make sure your assumptions are met. And that's what you get from the ANOVA Jamovi output.